Hello and welcome to Unsupervised Thinking, a podcast on neuroscience, artificial intelligence, and science more broadly. We are a group of computational neuroscientists. I'm Grace.
I'm Connor.
And I'm Josh.
And the topic for this episode is functional magnetic resonance imaging, or fMRI, and kind of more particularly how to analyze and think about data that you can collect using fMRI. So basically the outline for this episode is that we will first go over the basics of fMRI, the science behind it, what it's measuring, and how it measures it. And for that, we're going to draw a lot on a paper called Interpreting the Boldes Signal by Nikos Logothetis and Brian Wendell. And that's a 2004 paper that goes into some studies that cover kind of the connection between neural activity and the signal that's actually measured by fMRI. And then we'll get into more detail about, once you've collected this signal, once you've done experiments with fMRI, what do you do with that data? And we'll talk a little bit about historical approaches to that data, and then kind of the frontier, what people are doing now and what they'll probably be doing with fMRI data in the future. And for that, we read Computational Approaches to fMRI Analysis. This was part of a special Nature Neuroscience issue devoted to neuroimaging that came out recently in March of 2017. And the first author on that is Jonathan Cohen. And then after we go through those kinds of ways of analyzing the data, we're going to talk a little bit about kind of a broader question of how to think about when we record neural signals or some kind of signal from a brain area, how do we think about that signal? If we're interpreting in a way that says, can we as experimenters find information in it, is that actually relevant to how the brain thinks about the signal? So for that, we read Is Neuroimaging Measuring Information in the Brain? And that is a paper from 2016. The first author is Lee DeWitt in the Psychonomic Bulletin and Review. And that is a new word for me, psychonomic. So let's start with the basics of fMRI.
One thing that I guess it's somewhat worth keeping in mind is that this technique sort of came out of like a medical tool development. Maybe that's obvious, but I think thinking about this tool as something that people figured out how to use. They're sort of co-opting it from it originally being a medical tool is kind of where its origin is. So MRI.
Do you mean, MRI. So MRI is just magnetic resonance imaging, and that came before fMRI, which is the functional version.
Yeah, the functional version is just basically collecting data at like a faster sampling rate, right? Or am I...
Well, I think it's also, so let's get into what MRI is. So basically, if you are getting an MRI and maybe you have gotten one on various parts of your body, or if you had a head injury, you might have gotten one, just a regular MRI, you're put into a magnetic field, and that causes these dipoles that exist in the nuclei of hydrogen atoms that are in water molecules in your tissue. They kind of become aligned with the magnetic field, and then the machine delivers a pulse of radio frequencies that kind of knocks these things out of alignment, and then there's basically a signal that can be detected as they relax back into alignment. And that's kind of what's being measured, and different tissues will have different properties about how they get knocked out of alignment and fall back into alignment.
And the main thing is you just basically get like a grayscale image of the tissue, because different tissue will basically, they have more or less water molecules, or you can basically, So bone is like one color and soft tissue is a different color. And so you basically get like an image of the soft tissue of the body.
Yeah, so medically it allows for inspection of damage to soft tissues, whereas something like X-ray, is looking at bone but can't give you these soft tissues.
Because the X-rays just go right through soft tissue.
So that's the origin of MRI, or the origin of its use in the medical field. And then functional MRI is specifically looking at hemoglobin in the blood, and in particular, oxygenated hemoglobin has kind of a different signal than if the hemoglobin has been deoxygenated, meaning the oxygen has been kind of released from the blood into the tissue. And so fMRI can measure levels of oxygenated blood or changes in levels of oxygenation in the blood.
So, but it turns out, right, you would see this in a normal MRI. But I guess the thing about normal MRI is that it's kind of done somewhat slowly. So, like, you basically collect data from the image, like the person has to sit still, and you collect data for like a minute or something and average it, or many seconds. Whereas the big deal with fMRI is that you're taking a bunch of images of the brain at a relatively fast timescale, like once every second or once every 2 seconds or something like this. And so you're only averaging the data that you collect over a short period of time, like the 1 second between frames, between pictures of the brain. And so you can see, that the sort of, there's basically very small changes on the order of like 1 to 2%, right? Very small changes in the grayscale images, basically, that correspond to this. It was observed that these very small changes basically correlate with blood oxygenation.
Yeah, and that was observed by Ogawa, was the researcher. The fact that you can pick up blood changes with MRI. I think one of the differences between structural and functional MRI is the timing and the amplitude of the radio frequency pulses that you send. I think that's like a different setting on the machine if you want to do functional versus structural.
Yeah, but like conceptually, you're basically just taking there's obviously like technical details that go into this, but at like a high level, you're basically just taking pictures of the brain at a much shorter time scale than you are for normal MRI.
Sure. And the reason that the blood flow signal is something that we're talking about in neuroscience, I mean, you can imagine, okay, you can measure blood. What does that have to do with the brain? It just turns out that different areas of the brain, if there's a lot of neural activity going on there, will have an influx of oxygenated blood to them. The details of that, kind of the timing of it and what causes the blood inflow and how much and all of that is not super well known, but that's the basis of the idea that you can use this blood flow signal to tell you about neural activity in an area. But yeah, as you were saying, there is like compared to structural MRI, functional is working on a faster time scale, but compared to other measures of neural activity, it's working on a kind of slow time scale. So you may have neural activity changes on the time scale of like a couple hundred milliseconds, but that could show up in this, what's called the BOLD signal, the blood oxygen level dependent signal that people look at when they're looking at fMRI. Those changes could show up as kind of on the order of several seconds. And so there's not a good way of getting fine temporal resolution using fMRI, but the spatial resolution, meaning, if you're looking across the brain, how detailed are these different blood levels on the spatial level, is considered pretty decent. It's like a 2 millimeter, scale of spatial resolution.
So I guess we could talk about some of at a high level, this sort of sounds approximately reasonable, but you could imagine that it's got a lot of caveats and so on. So I guess in this logothetis paper, they discuss a few of the complications that arise. I mean, there's like it's extremely complicated. The mapping from, in some way, what we're interested in neuroscience is like, what are neurons doing for the most part? So we want to kind of be able to understand how neural signals are, what the signals are in that neurons are kind of producing, and how they interact and how they lead to how they correlate with different kind of behaviors or sensory signals and these kinds of things. And so I guess one of the exciting things about fMRI, when people realized that you could measure this BOLD signal, was that you could potentially have human subjects doing quite complicated tasks or simple tasks, and you can interact with human beings. You can instruct them to do whatever you want, and you could potentially have some measure of activity all over the brain. Again, fairly slowly relative to other kinds of recording methods that you can use in animals, like recording single cell activity and stuff like this. So what are some of the problems that arise when you're just using this BOLD signal as a kind of proxy or correlate of neural activity?
Yeah, so I mean, as we kind of alluded to, this relationship between both the blood flow and what the machine is actually picking up, and also the blood flow to the neural activity, these are all complicated relationships. Particularly, this paper talks about experiments where they were actually able to, in monkeys, record on an electrophysical level, so like picking up neural signals or local field potentials, which is like the sum of a bunch of neural electrical activity. If you want to hear more about us being confused about what local field potentials are, you can listen to our neural oscillations episode. So they were able to have both of these measurements and say, how related are they? And part of the complication is that relationship varies by brain area, because different brain areas have different vasculature and levels of kind of how many blood vessels are reaching that area and the dynamics of them. Even kind of small scale things about the structure of the blood vessels can be important. Like they were talking about little sphincters in the blood vessels that control the flow, and that can mean that the blood flow is different in different areas depending on the number of those things. And so the vascular system in the brain is complicated. So that's one issue. There's also an issue of time scale. So looking at kind of a short stimulus and trying to understand the relationship between the neural signal and the blood signal can give you some information. But then if you try to extrapolate and predict the relationship for longer stimuli, it doesn't always hold. So there's kind of regional differences and there's time scale differences.
And I mean, I guess in the simplest fMRI models, it's worth keeping in mind that like the assumption being made when analyzing fMRI data, and I guess we'll get to this more later though, but the assumption being made is that a given brain area responds or has activity with like some time scale and like delay relative to like offset relative to like when the thing is happening. So like if a certain brain region is supposed to be sort of active in a certain, like in response to a certain thing or like during a certain computation, you assume that brain region responds the same way across time, but relative to other brain regions, it might have like some response profile, which is called the hemodynamic response function. And so like a given area could respond differently, like with a different blurriness basically, and a different like offset relative to other brain regions. But that brain region, that specific region, responds the same way across all of time, which, in this paper as well, they call linear time invariance, which is like a standard engineering term for basically meaning something responds the same way across time.
Yeah, so it's like if you give like a punctate impulse, you could see the response in this area, and it would be kind of slow and drawn out because that's the time scale of this. And the idea is that you should be able to characterize that slow, drawn out response and use that to predict that area's response in response to a lot of different types of input.
So I don't know, maybe we can just give a concrete example of ways in which fMRI clearly does work, because to me, like when I heard about fMRI at first, not knowing a lot about neuroscience, it seemed kind of like unclear that it would do anything. Like, if you just hear about it, you can imagine it would work well or it would work not at all.
So I mean, it's hard to know naively, and it's certainly a popular thing. Like in the media, people talk about it a lot.
Right. So I don't know. I thought like just some of the examples that they gave in this paper were useful, simple examples. So they talk about maps. So in visual cortex, so there's a kind of a big part of the cortex in lots of animals, but particularly in primates and humans, that is we think of as being kind of in some way dedicated to vision, although of course in reality it can be more complicated. And one way of describing responses in these visual areas is by talking about kind of response properties, like receptive field properties. So, you might have neurons in some area that respond particularly well to little dots of light in some part of your visual field, or you might have neurons that only respond when there's stimuli in like the top right or in the top left or something like that. So they have like spatial selectivity. Or you could have neurons that respond very well to moving objects but not to static objects. And if you try to map using lots of different kinds of techniques, including fMRI, the organization of these response properties across the surface of the brain, then for many different properties you see that they have kind of some structured organization. It's not like random. And so like one kind of simple, in a way, confirmation of fMRI techniques is that you can verify that the fMRI can accurately measure visual maps in monkeys by also using some more direct technique like electrophysiological recording from the monkey's brains, for example, and that works.
And you can also do things like more directly than fMRI look at blood oxygenation levels in animals. So like instead of using fMRI, there are other techniques for looking at blood oxygenation levels, and you can correlate blood oxygenation levels with neural activity and blood oxygenation levels with, for example, fMRI signal. And so like there's this many-way control where we kind of know that to some extent neural activity is correlated with blood oxygenation level and blood oxygenation level is correlated with fMRI in a strong enough way that you can actually confirm relatively robust features of the brain that have been characterized by other methods. And so that gives you like a strong baseline that like, yes, the fMRI is telling you something that is seemingly quite real about basic features of neural activity.
But something that is important that has been characterized by these authors is that the thing that is the neural signal when both things are measured at the same time, the part of the neural signal that seems to be most predictive of the bold response is the local field potential, which is this messy sum of inputs to the area and kind of recurrent connections happening in the area, but it's not necessarily the spiking activity of the cells in that area, which would be considered kind of the output. That's the message that area is sending on to somewhere else. And so it's important to know that that's the thing that the bold is tracking more so than the spiking output.
So like maybe more closely related to like the inputs to that brain region than the outputs? Is that the interpretation you would give that?
That's a simple way to think about it. And one of the examples that they give is that, so as Connor was saying, like there are certain cells that respond to motion, visual motion, and other ones that respond to static visual inputs and that kind of thing. And so using fMRI, because you can do this kind of brain-wide scan of regions, it's kind of a nice way to figure out, okay, what areas are responsive to what kind of things. And one way to check if an area is responsive to motion stimulus is to show one direction of motion for a long time until the brain is kind of adapted to it, and then switch the direction of motion. And areas that have motion sensitivity should have like kind of a strong response at the switch. And part of the reason you need to show the first motion stimulus is just you don't want to just look at areas that have kind of a response to any visual onset. You want to just say, okay, like we've adapted to the fact that there's a visual onset. Now I'm only going to change the motion of the visual stimulus and see what areas respond. And so through this method, they found or kind of confirmed an area called V5 or MT that has motion selectivity, and the individual neurons there do have motion selectivity. But according to fMRI, they also said that this other area, V4, has motion selectivity. And when you look at individual spiking in neurons in V4, you don't see motion selectivity. And the reason that fMRI kind of got this wrong, it's assumed, is because there are connections from V5, this motion selective area, to V4, the one that's not motion selective, but presumably those inputs aren't strong enough to make the cells in V4 actually motion selective. But fMRI is picking up kind of the input to that region. And so they have inputs that are maybe motion selective, but that's not what the cells in that region show selectivity to. So that was kind of like a false positive on the part of fMRI. But yeah, so otherwise, I mean, it's been used to do these like large scale scans of brain areas to see what is selective to what. And so I guess we should say the first human fMRI studies were in 1992, which is maybe surprising. You know, if you read popular science, you might think that fMRI is like the only way anyone has ever done neuroscience or ever will. But it's pretty recent, but it was used to help find things like the face patches, these areas of the brain that respond to faces. A lot of times an fMRI study will be used, or even fMRI on an individual animal will be used to locate certain areas, and then people will go in more precisely with electrodes and look at individual neurons. Yeah, but so another problem with it being more a measure of the input to an area is that if you have, for example, part of the brain that does some sort of computation where it compares two inputs and it's supposed to get it out, like the spiking activity of that area would give you something about a comparison between two inputs. That area might be getting a lot of input, but it might conclude, these are the same, and so I'm not going to have any output or something like that, depending on what the computation of that area is.
Can you give an example of that kind of computation? I'm not
Yeah, calculating any kind of difference. If I want to know if...
So I understand it at an abstract level, but I'm trying to think about like a specific example.
Those kind of decision-making tasks, right? People usually think of that where you have like how many dots are going left versus how many dots are going right. The usual simplified model is that you have MT encoding the left and right, two MT populations, brain area MT, and then a different brain area subtracting them, right? Doing the subtraction. So if they're very balanced and equal, you'd have a big input and a big fMRIs, a big bold signal in the comparison region, but you'd have little spiking in that region because the difference would be around zero, right?
And so even the region potentially that is actually sensitive would not be as active, or would it as well?
In that case, the actual spiking of that area would be, nothing if the two things are canceling each other out. But the bold signal would say that there's a lot. So it's not necessarily a problem if you know what the bold signal is doing. So it's important that it be characterized across different tasks, across different regions, across different timescales, so that in any given case, I'm looking at bold. What does bold mean in this circumstance? Then it can be used appropriately. I think there's a problem only if you just assume it means something all the time, and that's, not what it's actually giving you.
But I'm not saying that everyone does this, but if one kind of thinks about fMRI as reflecting this region is involved in something having to do with this signal that we've checked for in it, then that's like a fairly coarse. Clearly people are going to want to look for things more specific than that. But in the simplest case, you can just say, is this region kind of involved in this kind of thing?
Involved in is vague enough that it's fine.
Yeah.
Okay, so now that we know something about the basis of the bold signal, we can talk about the types of analysis that people have done with this. So I guess I don't know if we use the word voxels yet, but that just refers to kind of an individual unit of the bold signal, a small part of the brain. I think they say 3 millimeters squared.
Well, it's whatever size. So, depending on the settings of the way you're conducting the fMRI. So, basically, another thing that's, I guess, kind of worth knowing about is that, basically, when you use a scanner to conduct one of these experiments, the quality of the scanner, in some sense, I guess, is, has to do with the strength of the magnetic field. So, a more powerful scanner corresponds to it being, a stronger magnetic field that can be generated. And that allows you to get, in a given time scale, in the same amount of time, you can get a higher resolution image. So I do not know the details of this, but my understanding is that basically the strength of the magnet allows you in some way to get a higher resolution image. So voxels can be different sizes depending on both the strength of the magnet and based on the amount of time you're willing to average over to get that voxel, basically. So there's a little bit also of a trade-off between how much time you spend and how many voxels you could collect and the resolution. So you could also do things like if you only want to do a scan of a certain subregion of the brain, you can get often higher resolution both temporally and spatially than you could if you were scanning the whole brain, which sort of generally makes sense given that there's some limitations on the technology. So there are these trade-offs that people make when they're...
Yeah, I think a good design is considered one where maybe you do a first pass of the whole brain and you look for what would be called regions of interest that seem like they're involved in the task that you've been testing on. And then, importantly, in a separate group of people in data you've collected separately, you look at that region of interest more specifically. And then you can test out kind of the hypothesis that you generated from your full brain scan. So if you have all these voxel measurements, kind of the old school way was to go voxel by voxel, and you have basically two experimental conditions. One would be the actual experimental one, looking at some task of interest, and then some control task that is ideally as similar as possible to your task of interest, excluding the interesting part. So you would have, if you have maybe some task where people are, I don't know, doing some math equations, you're showing them math that they have to solve in their head. I think maybe for a control task, you would want to have them still looking at a screen, maybe a screen that has numbers on it, but they're doing some sort of search task, maybe like looking for a specific number, not actually doing mental math, because then you're controlling a lot of stuff, and you just want to know, what areas are involved in mental math.
And this, I guess, this kind of subtractive method like dates back to like PET and other kind of earlier forms of imaging, where you'd have people just in like two conditions and subtract them.
And so then you're just looking for voxels that have a difference between these two tasks. And then you can say maybe, okay, this voxel shows a difference in these two tasks, so maybe this area is involved in the task of mental math or whatever the task is.
It's like more or less active.
Yeah.
I guess it's also worth maybe stating, if this wasn't clear, like, I don't know if this is explicitly true, but like I guess a voxel is like a volumetric pixel. That's kind of what it stands for.
Yeah. It's a portmanteau. Is that the phrase for words being smashed together?
Something like that.
So you could do this voxel-wise thing. An issue there, especially if you're doing kind of brain-wide scans, is that you have a lot of voxels, and then you're doing a lot of comparisons, and in statistics, that's bad. If you do a lot of comparisons, you're likely to have false positives, and so you're supposed to do a correction to kind of the level of evidence that you require to say something is different. So it's supposed to be stricter levels of evidence to say that you found a difference in the case where you're doing a lot of different comparisons.
So the sort of standard statistics test is something like, I think this is likely to have been an interesting phenomena if the probability that I could have observed it by chance would have been like 1 in 20 or 1 in 100 or something like that. So there's some relatively low probability of the thing occurring. And I guess specifically when doing hundreds or thousands or tens of thousands of statistical tests, you're going to expect hundreds of things that like have a 1 in 100 probability or something like that. Right. And so the right thing to do in that setting from a statistical standpoint is to either do fewer tests by like a priori, like before you start doing the test, say, I'm only going to do this test and I'm only going to do this test, thereby like making it fewer tests that you conduct, so that you're not just like getting all of these random positives and then after the fact saying they were all of those random things were real phenomena, or saying that because I'm doing tens of thousands of tests, I expect like a much higher standard of evidence. The problem with that latter thing from some perspective is that like you end up being like really conservative. If the effect size that you're expecting to see is somewhat small, you basically will miss the phenomenon, because you'll say like, well, this thing had like a 1 in 1,000 chance of happening, but I did thousands of tests, so I can't trust that this was not just random. But, essentially you would need to see something that's like, oh, this looks like it's 1 in 10 million chance of happening for you to say that it's significant if you're doing tens of thousands of tests. So there's this trade-off between you might miss things if you're too conservative that are really there. But if you're insufficiently conservative, you're going to assert that like lots of things are true that are not in fact true. So this is kind of a trade-off that people doing these kinds of data analysis things have to kind of dance. And I think given that there's sort of judgment and discretion in this level of conservatism, there's sort of risk that people will err on the side of overly optimistically interpreting things.
That's a political way to put it, overly optimistically interpreting. But yeah, so to help counter those kinds of problems, something that you can do is incorporate priors into your data. So you might assume that nearby voxels should behave somewhat similarly. So if you just see kind of like a random scattering of voxels that show significant differences, that might just be random. If you see many voxels in the same small region that are showing significant differences, then maybe you're more likely to be onto something there.
So the only, and this is true, and this is a way that people think about it. It's like if a whole bunch of neighboring voxels pass the same statistical test, then it's probably real. The only problem with that is, and I mean, that's like broad strokes, that's like a very reasonable thing to do. But the one kind of, it's worth keeping in mind that there's some interesting context, which is like, okay, as a human who's actually in the scanner, you're going to move a little bit. And so there are artifacts introduced into the data collection by the person moving around. So like, in fact, neighboring voxels are actually slightly artificially correlated, like sharing the same kind of structure, because the voxels aren't cleanly isolated. Like the person moves a little bit, so two neighboring voxels are actually like you're vibrating between them while you collect the data. So there's like messiness where like there's basically smearing or blurring of the data as it's collected. Now people try to correct for motion artifacts. Like people try to say this person, you can like infer that this person was moving by looking at like where their skull is and things like this. But that's these methods are essentially and where their brain is. And these methods are like a bit imperfect. So, and then also when people do analyze the data, they often add blurring to it as well. So there's sort of lots of complexity in terms of artifacts that can be introduced and blurring, and then also the fact that blood vessels are the things that cause the blood oxygenation level, and blood vessels might be larger than voxels in some cases. So like you might see a correlation across multiple neighboring voxels, but it's because there's a large blood vessel that is essentially the cause of both of those neighboring voxels reflecting some signal. So it's not as though like you can get down to an arbitrarily high level of precision.
And so these kinds of problems, about like motion correction and that kind of stuff, a lot of this is handled by these pre-processing toolboxes that many labs use. So these are software packages that are just kind of part of doing fMRI experiments. And they also can do, voxel-wise comparisons or any other type of analysis usually. And so this is generally considered a good thing that there's standardized methods being used across labs. But there was a paper last year that claimed that I think it was like 3 of the major packages had some errors in it where it was returning false positives at like an insanely high rate. And so they claimed that this kind of put in jeopardy a bunch of fMRI papers because so many labs use the same software systems. And then if there's an error in one of them, then it's contaminating all kinds of research. So that's the balance between standardized science and, if everyone wrote their own, then not all the labs would be affected.
But there'd be probably lots of distributed bugs that are different in everyone else's.
Bugs are different than you can average over them.
Sure. That's the right way of thinking.
Or there's the old eggs in baskets analogy, which...
Don't put all of your eggs in one basket.
Yeah, but then like if you have all of your eggs in different baskets, those are all the same kind of error, right? Dropping the egg from the basket. So averaging doesn't work when you have eggs in baskets. You know what I'm saying?
I think I've lost the thread of this
doesn't
comparison.
Matter. It's not central. It's not central to the discussion.
Returning to the thrust of this conversation, I guess they talk about lots of approaches in this paper. Some of it's sort of a review. I mean, basically, the standard methods that people use to analyze fMRI data consist basically of saying, like, I think this signal exists in this brain region or in a brain region. Let me do something like linear regression. Let me check to see if the brain signal looks like this signal. And try that maybe on each of the voxels independently or each ROI independently. And then for the brain regions that do look like this signal that you thought, then you would say, okay, that brain region is like sort of involved. That's like the most standard. And so like an example of this would be like, okay, the screen will be black and white, and the screen will like flicker from black to white or white to black at like some slow rate. And then like maybe you would expect some visual area like much of the visual cortex, would just basically have activity that looks like the flickering of that screen. So you would just basically perform linear regression between the color of the screen, like which is white or black at any given time, and every voxel in the brain. And you would see that not all brain regions are correlated with the flickering of that screen. And but brain regions that that are involved in vision are correlated with that, and maybe some other ones as well, because it's like such a salient phenomena. And then you could imagine generalizing this kind of logic to like many other kinds of things. So instead of it being like the flickering of the screen, you would show different kinds of images, and you would say when a certain kind of image, like a face, is present versus a non-face image, only certain brain regions involved in faces will have activity. So you can basically correlate the activity between that. Is the face present and is the brain region active? And you could do this for even more abstract things. Like maybe you'd have a task where you expect a person to be learning only during certain parts of the task, and at other parts of the task they wouldn't be learning, or something like this. And you would say, like, brain regions that have activity correlated with the times in the task when learning is taking place are involved in learning in this task. And so you can kind of generalize this to very abstract signals that are not just raw sensory inputs. And so that basically constitutes the sort of simplest way of analyzing fMRI data.
And so to do the more abstract things, like when will learning happen or something like that, usually requires that you have some sort of model of what you think is going on internally that has these like latent variables that you can say, well, in my model, this value changes this way over time as the person does this task. I could look for a signal that changes that way in the brain as well. And then I could say, oh, maybe the brain is implementing this model that I have of learning or of some other cognitive procedure or something like that. And you can do this in the individual voxel way, but, you know, the hip thing is to do multi-voxel pattern analysis, which just means that, you know, instead of saying, does the activity of this one voxel, you know, correlate with an input in some way, like a visual input, you can ask, if I look at a pattern across voxels, the activity of many different voxels in different conditions, can I find some pattern in that? Can I say the voxels look this way during one type of visual input and they look another way during a different type of visual input? So you're looking across voxels.
You're essentially looking, like maybe there's like some distributed pattern. And in some ways this is related. Like if you looked at it at an individual voxel level, you would expect to see all of the voxels that are relevant, you would see stuff in. Here, it's more like you're trying to sort of, in some sense, decode what the brain has, is going on in the brain. So like instead of saying, does this voxel relate to this signal, you say, from all of the voxels or from all of the voxels in some brain region, can I like predict something about my experiment or about the task?
Yeah, so usually the form this actually takes is that you do an experiment, you collect voxel data in different conditions, say showing images of faces versus showing images of scenes, and you train a classifier, usually something like logistic regression. And so you say, can I take this voxel information, I have the voxel information, I have what image was shown at that time, I can train a classifier to learn to say these voxel patterns relate to this kind of image, this other pattern relates to a different kind of image, and then I can show a person new images of the same categories and see if I can correctly predict just by looking at the voxels, like kind of what image they're looking at. And so you've trained a classifier on your voxel patterns, and then you test it using newly collected voxel data. And this, in some places, will be referred to as mind reading, which is a little generous. But this is a certain style of analysis that people do to show that in this brain region there exists the information about, say, the identity of the image that's being shown. I can read it out from this brain region with this classifier that I trained. Importantly, you generally have to train the classifier on an individual person. You can't, there's not, as of yet, that I know of, good ways to say, you know, I have voxel data from this person and I'm going to extrapolate
Well, so there's a little bit of that. So there's a thing they talk about as well.
The shared something?
Shared response modeling. And there's basically some subset of fMRI experiments where you can see that the simplest version of this would be like you do your per voxel analysis on each subject, and then you like average the brains across subjects. And you might see that some things that you've looked at hold true on an individual subject level, and even after averaging across subjects, you see something. And there's more complicated ways of doing it. It doesn't just have to be averaging. But you basically can see something like this phenomena is robust across individuals. And then you could imagine that a classifier would hold across individuals to some extent. Like if it were two very different things where like you would expect across individuals, totally different brain regions would be involved. You would expect that like the classifier would still work. Or maybe, I'm not like so very up on this, but, maybe there are some phenomena that are like sufficiently robust across individuals that even if it's kind of the same thing, like same general category, like different visual kind of things, you would still expect the classifier to have.
The issue is getting kind of a voxel to voxel lineup between two different people. And it's an area of research, and it's interesting to think about is how do you compare two different brains. I mean, the large scale anatomy is not so difficult. There are clear landmarks on the brain that people kind of line up and say this is this region, that's another region. But voxel by voxel is hard, and I think a lot of the attempts to go across individuals has to reduce the voxels into some lower dimensional space that they can kind of claim is common to both brains. So doing the multi-voxel stuff allows for maybe picking up signals that are harder to see in individual voxels. So an example is orientation tuning in the visual cortex. We know that the early areas of the visual cortex, individual neurons respond to different orientations of lines, to different edges, and but they generally have similar responses. Cells that are near each other prefer similar orientations, and the voxels are large enough that they're averaging over a lot of the different orientations, and so you don't necessarily get voxels that prefer individual orientations, even if all of their component neurons have their own preferences. So you can get maybe just small differences in the voxels, but if you look at multiple voxels, you can then have enough information to decode orientation.
Do you guys know of any very good fMRI papers?
Really?
That's my question.
That's your question?
Yeah. I just realized if I was listening to this, I'd be like, what are some really good, like what does fMRI do? Like Jack Gallant has that thing where he can decode complicated stuff, right? You can kind of decode what movies people are watching, like aspects of movies people are watching.
So in some sense, there's like two, like probably more, but like two broad categories of things people will try to do with fMRI. And one is like do science with it and try to figure out what the brain is doing. And the other is like do something like brain reading or like engineering, where you're trying to like decode what people are feeling or thinking by looking at their brain. And so there are things where people try to like reconstruct what people are looking at. And as based on conversations I've had with people involved in this kind of research, like one of the background goals would be do something like, if someone's dreaming, could you reconstruct what their dream is? And the way you might like think about going, like endeavoring on this is start by giving them, like forcing them to watch movies while you record their brain. And then while they're asleep, if you've got a good enough thing that can decode the movies they're watching while they're awake, and you've learned that in a sort of supervised way by like comparing what you showed them to what their brain was doing, and you can sort of do it there. Maybe when they're dreaming, you'd be able to decode what they're dreaming.
Or even if they're awake and imagining something.
Sure, yeah. And so there's lots of variants of this that basically constitute some form of mind reading, where maybe part of the way you've trained the process by which you're going to do the mind reading is by like supervised data, where you knew what you were showing them and you forced them to watch it, or they volunteered to watch it. In no case in experiments
you're just jumping ahead to the dystopian future.
Well, no, like that. Yeah. The Clockwork Orange thing is like you take like a prisoner and force them to watch some video. I mean, this is talked about in neuroethics, right? Like, what if, for example, you could force a person to be scanned and show them images or movies of a walkthrough of a crime scene and be able to detect based on their neural activity while they're watching that if they'd been in that place before.
It's like fMRI lie detectors, like that kind of thing.
Yeah. It's a generalized form of lie detector, but it's somehow it's different than like in a normal lie detector. You ask a person a question and you can assess whether or not they're lying. Here, you could sort of force information out of people just by scanning them while like providing them stimuli that may evoke expected responses. And so like that's a kind of interesting and weird variant of this. And I guess there's like a field of like neurolaw, which is like hypothetically if these neuroscience-y things happen and get like used, like if courts decided to do that, would it be admissible? In part, constitutionally, like you can't be forced to incriminate yourself.
But yeah, but this is all, or at least most of what you were talking about is a generalization of the idea that you train a classifier on some data and then use it to predict things with new data, with new voxel patterns.
Sure. Yeah, of course. It's worth keeping in mind, though, like sometimes when people say they can classify something, they can do so only with like still relatively low accuracy. It's just much greater than chance.
Right. And there's also those things about deep networks. People fit those models. They do that with fMRI sometimes too, right?
Yeah. So basically, there is another type of analysis that isn't really training a classifier, and it's a representational similarity analysis, which means that if you show a bunch of different type of images, maybe a bunch of different people's faces and different scenes of different locations, something like that, and you look at the voxel responses, you can ask how similar is the response to two different images. And you can create this matrix of similarity between two different images. And usually if you do this, you'll probably see in certain brain regions, like, all the different pictures of a dog show up kind of similar in the voxel patterns, and they are different than pictures of faces. And so you can get this matrix that says, like, according to this representation, how similar are these different images? And what people have done, especially Nico Kriegescore, is to make those matrices for fMRI with fMRI data, and then also make those matrices for models of the visual system. For example, deep convolutional neural networks that have been trained to classify images. And so what they've shown is that you can create similar representations in these models. If they're trained the right way on the right kind of images, you can show that these matrices of similarity are similar between the brain and the model. And so this is a way of measuring how good is my model at representing things the way that the brain does. So that's a different type of analysis that I think is becoming even more popular now, with deep learning being popular. So another thing that they were talking about is real-time analysis. So the idea that you are processing the fMRI data as it's coming in during the experiment and using that to influence the experiment. So either to try to get people to control part of their neural activity. If you give them a feedback saying this part of your brain is becoming active, like make it more active or turn it down. Even if they don't explicitly know how to do that, they can, through trial and error, kind of figure out ways to control it. To be honest, I don't know what the scientific conclusions of those kinds of studies are. Just that certain brain areas can be controlled more than others, I think, maybe is something that's looked at. It's also just cool.
I mean, I think neurofeedback generally, which is what this is called, like, so you have any kind of closed loop setting. Okay, so in the EEG literature, I think it's maybe a little bit more straightforward. So like there are certain, for example, signals in EEG that are like correlated with wakefulness or like rest or being alert. And so like very coarsely, if you're provided some processed signal from the EEG that's like, are you resting or something, or are you relaxed? And maybe you're anxious. You could do like neurofeedback to like try to calm your brain or something like this. Now, I'm not so critical of this if there's a clinical use for it, but like I guess it feels a bit fuzzy to me.
Yeah, I mean, it's EEG, But I do remember at the Society for Neuroscience conference, there was like a vendor set up who had this game where you had to try to relax yourself. And it was like a competition, like whoever's EEG signal was most relaxed, there was like a ball on a table or something, and it would go towards them so you knew who was winning. And that's a fun game because if you start to win, you get excited, but then you'll lose. So that's a good use of it.
So briefly, a few years back, there was like a little bit of a commercial buzz around this kind of neurofeedback, EEG neurofeedback stuff with like commercial headsets that like had, you know, some sort of small number of EEG electrodes. And I remember there was one that was like related to Star Wars. It was like, you know, using the force to like make some ball levitate, which was really just, there was like basically a fan that made a ball float, like a ping pong ball float. And it floated more if you were like, you know, in some EEG related state, like being, you know, alert and restful or something like that.
The other thing you can use the real-time stuff for is to kind of try to find maybe the optimal stimulus for an area. If you can see on the fly that an area is responding to something, you can kind of show the person or have the person do something that's even more in that direction and see if it makes that area even more active, that kind of thing. So that's another use for the online analysis. So then our final topic is a little more, I guess, on the philosophical side of what are we actually doing if we say that we can decode certain information from a brain area, or if we can train classifiers to decode information. So this was a paper that was kind of just trying to, I guess the central point of it was to say that it seems like a lot of these studies that look at brain region involvement are treating the experimenter as the receiver of the information. And the question is, can the experimenter read out information and use information that exists in the signals that they've recorded? But the question should be, can other brain areas access this information? Or, you know, can we say anything about the mechanisms by which other brain regions might access this?
And then, like, there's all this thing of, like, information, things being encoded in a certain way, and, like, can you access them and whatnot? Can I access them by looking at your brain, and can some part of your brain access the information being encoded in some other part of your brain? Like, in the end, what we actually care about is kind of how complex behaviors arise out of, like, the physical properties and, like, dynamics of the brain. It's already like a massive simplification.
So, I mean, one way of maybe thinking about this, though, is like, I mean, they acknowledge this in this discussion, that like some kinds of, like this classification we had between like the more engineering decoding kind of stuff versus science. For science, you want to know what the brain is using the signals that you see in the fMRI for. But like, maybe there are engineering purposes, like mind reading kind of purposes, where you don't care if the brain can do it. You just care if you can do it.
Or like medical diagnosis.
Sure, of course.
Yeah, there's plenty of reasons.
Yeah, which is a kind of like engineering application in some generalized sense. This like multi-voxel pattern analysis kind of approach, if you look at the whole brain and kind of, in some hypothetical sense, only using the entirety of the brain, you can classify something, like what the person was thinking. But it requires the whole brain. It's improbable, perhaps, that a specific brain region has access to the information that the entire brain has when you're reading it. Like when you as the experimenter are looking at the entire brain and require the entire brain to perform some classification, maybe there aren't brain regions that would have access to equivalent amounts of information.
And then there's also another kind of clear distinction which has come up recently, not just in fMRI, but also in studies of decoding information from recorded spiking activity of neurons in monkeys. Because the question of like, can you classify the stimulus at all, versus in what way is that information encoded? You know, so that's another kind of concrete thing. So, for example, you know, it might be that with a very complicated classifier, in some situations, you can read out what an animal or a human being was looking at, say, but if you're using simpler classifiers, it might be that only in certain situations can you read it out. So like concrete example of this, not from fMRI actually, but from a study that used direct neural recordings in monkeys, is you can try to read out some stimulus that the monkey was looking at. And there was a study that found that using a certain classifier, you could only read out information about the stimulus when the monkey had performed well in some way. But if you used a more complicated classifier, you could always read out the stimulus. So that kind of hints.
So even when the monkey made a mistake on that particular trial, you're saying.
So if the monkey made a mistake, you might think, oh, well, he didn't know what the stimulus was. But it turns out that if you use a very complicated classifier, you can read out from his neural activity what the stimulus was. So somehow the information about the stimulus is in his brain.
That's
but the fact
cool.
that he made a mistake
That's
kind of
cool.
hints at the idea that he couldn't access the information. And so the idea is that, the information is encoded in like too complicated a way for him to use, even though you can decode it from his brain, basically.
Which is kind of strange, almost philosophically, right? I mean, this kind of gets at the heart of this distinction between what an experimenter can read out and what the brain is capable of accessing. I mean, I guess we sort of naively think that we have access to most of the information that our brain has. I guess modular, like some subconscious stuff. But it would be weird, kind of, if like subconsciously we could solve problems that we weren't aware we could solve. Sort of. I mean, that's kind of weird. I mean, it's not really subconscious because I don't want to like elide over the distinction between like the neural activity somewhere in your brain having it and your like mind having it or something like that. It's not clear that like, yeah. I mean, it could be like some very low level, almost sensory area in the brain has access to this information and not like any cognitively relevant area.
I mean, that's just like, it can become kind of trivial if you think of it as like, if you were recording all of the neurons in the monkey's retina, then like obviously you would expect to be able to read out what he's looking at always. But he may be, for example, just ignoring it or something, right? Yeah.
The fact that the brain may have some signals that are kind of close to or analogous to the recording of a camera that are like not adequately processed, you're seeing directly the limitations of the animal. Which is not something you tend to think about. You tend to think about like by the time it gets to the brain, it's stuff that the animal should be capable of processing. But like animals are imperfect, and so the fact that when you're observing fully their brain, I mean, we're not observing fully their brain, but like if you were hypothetically fully observing the brain, you'd be able to perform closer to the way you could perform with like raw inputs as if you were like a perfect machine, optimally processing the inputs.
Yeah, it's kind of interesting. You can think of it also as like, I mean, you can also look at it in a slightly different way, which is that the brain is taking in so much sensory information all the time. A part of its active, if you want, goal-driven kind of task, like part of what it has to do is to filter out and ignore lots of information. So you can have this image of the brain as like having all this extraneous crap bouncing around and like, reverberating through it, and there's some complex process of trying to figure out which is relevant, which is not.
Yeah, and so I feel like so there are examples of this on the neural level, even when you have individual neuron data, but it's kind of, the issues can be even starker with fMRI because you're really measuring something that we don't consider the thing that encodes information directly in the brain. Like, we don't think that neurons are reading out the blood flow signals, even though that's what fMRI is picking up. So there's even more opportunity for analyzing a signal that other neurons don't really have access to. And then the other way is that you don't find the information in an area, but in fact it is there because you're averaging over a bunch of neurons or something like that. And so that's, I mean, even a more obvious problem for fMRI in particular is that you cannot really confidently say, oh, this brain region doesn't have this information, because you're not looking at what would be assumed the right scale to be able to say that. We assume that spikes are the important thing, action potentials in individual neurons, to access it. So I guess that's kind of the current state of fMRI now. A little more interesting in terms of the analyses that can be done due to computational power and algorithmic developments.
I'm thinking.
Till next time. Hey, if you're still listening to this, you must really like us. So how about you go to iTunes or Stitcher and rate the podcast, give us some feedback. You can also go to our website, unsupervisedthinkingpodcast.blogspot.com. You can comment on different episodes, or you could give us ideas for new topics you want to hear about. We would love to hear from you. Thanks.